Deleting APOE4 from just one cell type reversed memory loss in mice. This creates massive implications for prevention. We usually think of Alzheimer's as something written in our DNA, but new science shows your story isn't set in stone. It's shaped by how you live, how your brain gets oxygen, and how your immune system responds to stress. I'm Dr. Kevin Tran, and in this episode, we're breaking down 4 eye-opening discoveries straight from the Alzheimer's Association International Conference on APOE and Lipid Biology held in March 2025, so this is extremely fresh research. We'll explore how APOE4 interacts with your environment, your blood vessels, and your brain's immune system, and what you can actually do about it. So whether you're tracking biomarkers, tweaking your habits, or just starting to learn about it, this video will help you make smarter decisions to stay sharp, hopefully, for life. When we talk about Alzheimer's, we usually jump straight to genes and brain scans. But Dr. Naomi Ferreira's work forces us to zoom out to the life circumstances that shape how your brain ages. Her team asked, what if Alzheimer's risk isn't just written in our DNA, but also in our daily life? Things like education level, financial stability, and whether or not you have close relationships. And the result? Well, APOE4 might load the gun, but it's your environment that pulls the trigger.
Social determinants of health like low educational level, low socioeconomic status, and social isolation can negatively impact cognitive reserve, increasing dementia risk.
So those researchers analyzed over 1,000 brains in Brazil, which is a country with high social diversity and deep inequality. This wasn't some idealized lab cohort. It was a real-world snapshot, people who had lived vastly different lives in terms of opportunity, connection, and stress. They measured classic Alzheimer's markers, amyloid plaques, tau tangles, neuronal loss, but they didn't stop there. They layered in powerful context. So what levels of education did this person reach? Did they live alone or with a community? Were they economically secure or were they in chronic stress? And then came the surprise.
The interaction of social determinants of health on the association of APOE4 with AD pathology was not significant.
Here's what that means. APOE4 still caused plaque buildup regardless of life circumstances. So no, social support won't stop the biology from unfolding, but that's not the full story.
However, the interaction of social determinants of health on the association of AD pathology with cognitive abilities was significant. The association of AD pathology with poor cognitive abilities was stronger in the unfavorable compared to the favorable social determinants of health profile.
In simple terms, 2 people could have the same amount of brain damage, but one stays sharp while the other declines rapidly. Why? Because cognitive resilience isn't just about what happens in the brain. It's about what supports your brain. Education, mental stimulation, meaningful relationships, these things build buffer circuits that let your brain reroute any damaged areas. It compensates for it. It's like having multiple highways when one road is under construction. Well, you take another one. So if you carry APOE4, this is one of the most empowering findings in the field because while you can't change your gene, you can shape your brain's environment every single day. You can keep learning, you can stay socially connected, you can cultivate curiosity and purpose, like watching these videos. And I suggest you subscribe to this YouTube channel so you don't miss any of my future videos on APOE4 and how to defeat Alzheimer's. All right, moving on to the second part of this conference. Most people think of blood vessels as passive pipes, just plumbing for the brain. But that's not the case. They are actually smart, responsive systems constantly adjusting blood flow based on what your neurons need. And APOE4, while it hijacks those control settings, especially in the support cell that keep blood vessels flexible and responsive. Dr. Yasuteru Inoue's team zoomed in one critical cell type, the mural cell. Those move muscle-like wrappers around your brain's smallest vessels. Think of them like the hands on a faucet. If that hand is too tight and blood flow gets choked. If it's too loose and then the blood pressure drops. So the researchers asked a simple but powerful question. What happens if we delete APOE4 only in these mural cells?
APOE in vascular mural cells have a critical role in maintaining the cerebral blood flow.
So here's what you need to know. Mural cells aren't just structural, they are dynamic regulators of blood flow in the brain. especially important when neurons are firing and demanding more oxygen. In a healthy system, they tighten or relax like valves, ensuring precise delivery. But when APOE4 is present, it seems to scramble that feedback loop. Blood flow becomes less responsive, oxygen delivery gets delayed, and over time, this contributes to cognitive fatigue and early damage. So Inouye's team tried a radical intervention. They deleted APOE4 only from those vascular support cells, leaving the rest of the brain untouched. So what happened?
E4 deletion resulted in the improvement of the spatial memory retention assessed by Morris Walter Mays.
Well, if we're talking about it, it's usually positive result. Memory improved without any drugs, without overhauling the immune system or even removing the plaques. Just by removing APOE4 from a single overlooked cell type, they restored more normal blood flow, and that translated into better spatial memory in mice. This wasn't about boosting performance, it's about removing the friction that was getting in the way of normal brain function. So this study reveals something very important. APOE4 isn't universally bad, it's the context where it is, the tissue, the timing, the interaction that determines its effect. So in the Phoenix community, we call that targeted resilience. We don't do shotgun approaches. It's not like, you carry APOE4, just take this supplement and you're done. Instead, we focus on where the damage starts and how to restore function from the ground up. So if you optimize your vascular responsiveness through exercise, for example, nitric oxide support, and mitochondrial health, you improve dramatically your cognitive performance. Exploring biomarkers-guided intervention so we know where APOE4 is active in your system is extremely important, and designing the habits that improve neurovascular coupling, the alignment of blood flow and the brain activity is also critical for your brain health. Because if one small change in one small cell type can restore memory in mice, just imagine what the right interventions could do to you. The third person who talked in that conference was Dr. Crystal Lang. When we think about Alzheimer's, we usually picture neurons and memory loss. But what if the earliest warning signs aren't in the brain cells, but in the blood vessels feeding them? So it follows on the previous insight. Dr. Crystal Lang research shows that vascular damage begins years before symptoms, and it starts earlier and more aggressively in APOE4 carriers. We're not talking about big dramatic strokes, we're talking about a slow, silent suffocation where tiny vessels degrade, oxygen delivery weakens, and the brain's repair system flickers out. Let's break that down.
APOE4 mice demonstrated reduced expression of VEGFR2 globally, with the signal expression at 12 to 14 months measuring far below expression levels for time point 1, 2 to 4 months. So this deficit was only observed with VEGFR2 marker in the APOE4 group and not seen at all with the VEGFR2 marker in the APOE3 group.
VEGFR2 is like the common switch for vascular repair. It responds to damage by telling the body, all right, we need new blood vessels here. Think of it like a construction foreman calling in workers and material to fix a leaky pipe. But in APOE4 mice, that foreman starts ghosting the job. By middle age, equivalent to, let's say, your 40s or 50s, the signal to repair the blood vessel fades, and the crew never show up. And when that signal is lost, the damage compounds.
There was a significant reduction in thalamic vascular density as well as thalamic tortuosity for the E4 group.
So now the blood vessel network itself starts to thin out. Vascular density drops, which means fewer pipes delivering nutrients to your brain, and tortuosity changes, which means that the vessels become less flexible and adaptive, so they're more rigid. A good analogy would be to think of it as a city slowly losing its plumbing infrastructure. Fewer pipes, more blockage, and no detour routes when the traffic gets bad. So the neurons are still all there, but now they're starving for oxygen and energy. And it doesn't stop there.
So briefly, to further understand molecular changes involved in the vascular pathology observed, I performed proteomic analyses on isolated microvessels, and the preliminary data identified significantly downregulated pathways involving VEGF and PDGF signaling, as well as upregulated mitochondrial dysfunction in APOE4 by 7 to 9 months.
So even before the visible vessel loss, molecular signals for blood vessels maintenance and repair, like VEGF and PDGF, are already fading. At the same time, mitochondria, the cell's power plant, begin to fail. And when your brain is both underfed and underpowered, you start to feel it. You feel it in fatigue, brain fog, slower mental recovery after stress and poor sleep. So these changes are subtle at first, but they are real. So what does it mean for you? So your blood vessels are your early warning system. In the Phoenix, we always focus on vascular health and mitochondrial resilience. And you can do things like tracking your VO2 max. So that's the oxygen your body can process, and any smartwatch or even a Oura ring can be able to track that. That's a vital metric for brain perfusion. You can also explore retinal vessel imaging, which mirrors the brain vasculature that gives you insight on the state of your brain health. And then you can watch endothelial markers like nitric oxide availability or arterial stiffness. No matter what, cognitive decline often starts with blood flow decline. And the sooner you spot that, the more power you have to change your trajectory. The next person that talked in that conference was Andrew Pearson on microglia. I feel like it's a topic we cover in a lot of different videos, and that shows how important microglia is for APOE4s and Alzheimer. Well, when it comes to brain health, microglia are both the guardians and the potential saboteurs. These immune cells patrol your brain, they clean up the debris, they repair damages, and they respond to threats. But in APOE4 brains, they don't always know when to stop. So Dr. Andrew Pearson's research shows that APOE4 rewires this cell into basically overzealous cleanup crew. They are actually making things worse. Let's walk through how this plays out.
By 6 months of age, we see that APOE4 vessels have an increased number of methoxyl amyloid positive vessels. And when we analyzed these vessels for A beta species, we saw that E4 vessels had significantly higher levels of insoluble A beta 40.
So 6 months of age is very early adulthood in a mouse. The blood vessels in APOE4 brains are already clogged with insoluble amyloid beta 40. This condition, known as cerebral amyloid angiopathy, is like plaque building up in your brain's capillaries. Instead of oxygen and nutrients flowing freely, you've got sticky gunk blocking the pipes. Now, normally the brain will call in its immune task force, the famous microglia, to handle the cleanup, but here's where APOE4 throws a wrench in the entire system.
We saw there was a much greater number of microglia around each plaque in E4 mice. And within this 70 micron ring we drew around each plaque domain, there's a much greater CD68 immunoreactivity, suggesting a hyperreactive phenotype of these microglia.
Rather than calmly clearing the damage, APOE4 microglia go into overdrive. They cluster tightly around the plaques, they ramp up inflammation, they start releasing reactive molecules that can harm surrounding tissue. It's like if you had firefighters flooding a room with so much water, they basically collapse the entire floor. So the intent is protection, but the outcome is destruction of everything there. This hyperactivity is measurable too. It's a higher CD68 expression, a marker of inflammation and overactivation. And here's the really wild part.
What we actually saw in the E3 mice is that depletion and repopulation actually reduced the amyloid burden around the vessels. And when we looked using the ELISA to measure insoluble species, we found that E3 mice, APOE A beta 40 was actually reduced. But while we didn't see the same effect in the APOE4 mice, we did see a big increase in markers of neurovascular coupling. We also saw Wnt beta-catenin signaling increases and also endothelial nitric oxide signaling.
Even when scientists wiped out the microglia entirely and allowed a fresh new generation to regrow, the APOE4 brain still didn't bounce back. That's a big deal. It suggests the damage isn't just from the microglia behavior, it's baked into the system. The environment they repopulate is already primed for dysfunction. It's as if the brain's immune software has a corrupted install file, and every time you reboot, it loads the same errors. So what can you do about it as an APOE4 carrier? Well, you can improve your sleep, which basically trains your microglia to behave more cleanly when you have proper deep sleep. You can have cold exposure and exercise, which helps regulate the immune tone. You can take polyphenols like resveratrol and EGCG, which can dampen chronic inflammation. And you can modulate your stress from doing breath work to HRV training. Because in APOE4, we're not trying to suppress the immune system. We're more like teaching it to respond wisely. Because in APOE4, a calmer immune system could be your best defense against brain breakdown. Across these 4 studies that we covered today, a powerful message is emerging, one that every APOE4 carrier needs to hear. Risk isn't rigid, it's responsive. We saw how. 1, social support and education can literally buffer the brain against damage, keeping people sharp even with significant Alzheimer's pathology. 2, targeted removal of APOE4 in vascular support cells restored memory in mice, not by adding something, but by removing friction at the source. 3, early vascular and mitochondrial breakdown sets the stage for cognitive decline years before symptoms and years before any diagnosis. And 4, microglia, the brain's immune crew, become overactive in APOE4, and that hypervigilance leaves lasting damage even after a full reset. And through it all, one truth becomes very clear. You are not your genes. You are the sum of your choices, your environment, your ancestry, your biology, your habits, all interacting in dynamic and often surprising ways. So at the Phoenix community, we don't just try to teach you the science, we help you apply it to your life. Because precision Alzheimer's prevention isn't about fear, it's about strategy, structure, and support, rooted in data that is adapted to you, because there is no one-size-fits-all here. So if you're an APOE4 carrier and ready to fight back, to experiment, to try, to thrive, apply to join us. The link is in the description below, and mention that you're coming from YouTube in the applications, because I read all of them personally, and I'll make sure that you get some extra love. If this video helped you, please share it. You might change the trajectory of someone that you love. I'm Dr. Kevin Tran. Thank you for watching, and then see you next time for another science-backed deep dive into the future of brain health. Bye.